So, next topic is the restriction endonucleases. These restriction endonucleases are enzymes. So they are coming under nucleases, enzyme called nucleases. The nucleases are enzymes that digest the nucleic acids. Nucleases are enzymes that digest nucleic acids. There are two types of nucleases: endonucleases and exonucleases. Endonucleases are enzymes that cut internal cuts in the nucleic acids. They cut at the internal points in the nucleic acids, but exonucleases cut the terminal ends of the nucleic acids. And some of the endonucleases are site specific. Some of the endonucleases are site specific. Site specific endonucleases are called restriction endonucleases. There are other type of endonucleases also that are not, they cut randomly, but there are certain endonucleases called restriction endonucleases. They are site specific or sequence specific, and these specific sites that are cut by restriction endonucleases are called restriction sites. These sites are called restriction sites, and hence these molecules or the endonucleases are referred to as molecular scissors. And these restriction endonucleases are produced by many prokaryotic organisms, microorganisms such as bacteria, and this was first discovered by Hamilton Smith and Daniel Nathans in 1970, and they got Nobel Prize for that. And different prokaryotes produce different types of endonucleases, restriction endonucleases, or some of the organisms produce many type of restriction endonucleases, restriction enzymes. Many organisms produce many type of restriction endonucleases. Same organism produce many type of restriction enzymes. And about the nomenclature of restriction endonucleases, you can see many type of restriction enzymes. So the source organism is Escherichia coli. So for naming a restriction enzyme, first the first letter of the genus is taken. Here first letter E is taken. Second is two letters of the genus will be taken. Two letters, CO is taken, so eco. And the next considering the strain of the bacteria, strain of the microorganism. Here the strain is RY13, so R is taken from that R, and this species or strain is producing only one type of restriction enzyme, so given the name 1, so eco R1. So restriction enzymes are named by using first letter of the genus, then first two letters of the species, and then the strain of the bacteria, and that will be suffixed. And when the strain produces more than one restriction enzyme, it is given as 3. Here Haemophilus influenzae, so Haemophilus H and influenzae IN, and its strain is D, and 3 means it produces three types of restriction enzymes, and this is identified by Roman numerals. And thus eco R1 is the restriction endonuclease produced by Escherichia coli belonging to the strain RY13. At present, the hundreds of different restriction endonucleases have been identified and characterized. So what is the biological function? Why these prokaryotes are producing this type of endonucleases? So the biological function of the restriction endonucleases is that they need to protect themselves from other microorganisms, other bacteria, other viruses. So to protect their genetic material for the of the prokaryotes from the invading foreign DNA such as bacteriophages, the restriction enzymes are produced by these bacteria.